Hello, let's start the introduction webinar for the AMO Webtrix. AMO Platform technology, we call it Webtrix as our brand name, which is synthetic microenvironment technology, mimics cellular microenvironment in human body. Amolifescience can engineer customizable in vivo-like microenvironment, and we provide 2D or 3D-like nano fibrous surface where key components such as ECM and growth factors, single or in combination, in the microenvironment are immobilized. How we engineer synthetic microenvironment. AMOLs use two kinds of our biomaterials from material science-based technologies of AMO Group. One is here mimetic protein library. It is core of our synthetic microenvironment technology. Our protein lines are recombinant proteins obtained by fusion of a bioactive peptide motif from proteins in human microenvironments such as ECM, with muscle adhesive protein. These proteins in our fusion protein library are coated on AMO Webtrix surface in order to mimic biochemical environment of human microenvironment. There are various tight extracellular matrix, growth factors, cytokines, cadherin and so on in our mimetic library. Many lines comes from ECM. We have collagen type 13 lines, laminin type 80 lines and fibronectin type 56 lines. Also, we have one more, very important and strong biomaterial, AMO nanofiber membrane in-house mass-produced by electrospinning technology. AMOLs can simplify nanofibrous substrate of in vivo substrate by AMO nanofiber. It means we can provide in vivo-like microenvironment and cells their behavior as in vivo environment. AMO nanofiber technology offers uniform-sized nanofibrous substrate to support self-renewal and proliferation of various stem cells including induced pluripotent stem cells. In-house mass production of nanofiber membrane enables to keep low variation in nanofiber diameter and topography and lot-to-lot consistency. To control diameter ranges from 100 to 1000 nanometers and membrane thickness finely and semi-transparency. Also biodegradable polymers can be used. In summarizing the things we have talked about above, AMO Webtrix platform technology provides the physical environment with AMO nanofiber membrane and biochemical environment with AMO mimetic protein peptide library to mimic human cellular microenvironment. Now AMOLs developed physically and biochemically defined substrate to mimic cellular microenvironment, and firstly, we commercialized defined cell culture surface as one of cell culture solutions, which is the first application. Also, we have been researching and developing other cell culture solutions such as cell culture media. And we have 3 more applications. Cell-based assays and cell expansion bioreactors have already been developed, and especially AMOLs cooperates to develop our bioreactor with Korean cell therapy companies. It would be possible to run CDMO business using our own bioreactors in the near future, and Korean cell therapy companies have used our cell culture surface products to maintain and expand their cells, not only for research but also for therapeutic use. The first commercial products of Webtrix are for stem cell R&D, especially pluripotent stem cell. Integrins play a crucial role in the self-renewal, proliferation and differentiation of pluripotent stem cell due to their ability to transfer signals from the ECM to the cell. Several key integrants for self-renewal and proliferation are alpha-V beta-3, alpha-2 beta-1, alpha-5 beta-1, and alpha-6 beta-1. Defined substrate that mediates the strong adhesion of HESCs and high PSCs via integrin alpha-V beta-3 or alpha-6 beta-1. AMO Webtrix products shows excellent performance for cell growth, proliferation. In the below, you may find that growth curves of human ESC on our Webtrix products are almost same to those on the leading products M and V. Also, AMO Webtrix product is pre-coated, user-friendly and ready to use. Off-the-shelf product, you can save your time by getting rid of the coating step, which is different from when you work with other competing products in the market which are substrate products for surface coating. And also, You can store our products for 3 years at room temperature, but most of other coating agents must store at 4 or 20, and so they need cold chain for delivery. Our primary concern is saving your valuable time. Now, I would like to introduce two kinds of series of Webtrix product line for PSC. First ones are pre-coated ready-to-use micro-well plate and tea flask. 6 to 96 well format available upon your request. Next one is bioreactor for large-scale production, as default. 10-layered growth surface for mass production of IPSC is available, and we can adjust the layer number in response to your needs or demand. Also, we can offer customizable substrate for your special application, and you can choose 2D flat surface or nanofiber surface. From here, details of Webtrix efficacy data in-house or from customers' evaluation for stem cell expansion will be represented in growth curve of HESC and HIPS in various media. Substrate effect on population doubling time showed Webtrix performance was comparable when compared to two leading products, M and V. In this in-house test, we used 1S and 2 IPS cell lines and 4 media, which are Stem Max, MTESR, TESR2 and Neuristem. Here you can find that morphology of culture cells on Webtrix surface in passage 12 was almost same to that in passage 1. In this slide, a compact, epithelioid morphology was observed, followed by the emergence of typical HESC colonies with tight morphology. The size and shape of colony formed on Webtrix surface were similar to those of cells cultured on leading products such as M and V. And markers of 2 human pluripotent stem cells, embryonic and induced, such as DAPI, October 4, SOX2 and MERGE, remained highly expressed for at least 12 passages, indicating cells maintained pluripotency through 12 passages. This is the results from customers' evaluation. Human embryonic stem cells were expanded and differentiated into midbrain dopaminergic neuron on AMO Webtrix PSC. Webtrix showed excellent performance, similar to C, based on expression of 2 markers, FOXA2 and LMX1A. For 3D cluster formation of mesenchymal stem cell for cell therapy, MSC expansion test was conducted on 2 types of Webtrix surface in comparison with C, and the company concluded Webtrix PSC showed similar or better performance and consider Webtrix for clinical grade MSC cell expansion. Commercial products and services utilizing AMO Webtrix technology are available now. We hope to meet you via our customer service. Thank you so much for your attention.